enzyme. But you guys kind of have a little bit of an idea because when we talked about fermentation, we did say fermentation starts with glycolysis. What that means is literally glucose is lysed into 2 smaller pieces, so that's why it has the word lysis in it. It's literally split into 2 smaller molecules. But what fermentation does not include is it does not include Krebs, and it does not include this electron transport chain. Krebs and electron transport chain is where glucose is broken down even further. It's oxidized all the way down to carbon dioxide gas and ATP, basically. But yeah, this is where you get all your ATP. Electron transport chain is really like the ATP making machine, and that's where the organisms really grab most of their ATP, not really from fermentation. But we did say the benefit of fermentation, though, is, what was the benefit? You can live without oxygen, at least. That's a big benefit, though. So there's not a lot of ATP to be made, but that's a big benefit if you can live without oxygen. So what we're saying here is that most organisms do use oxygen here at the end, but it doesn't necessarily have to be oxygen. Because they're, for example, like a lot of those facultative anaerobes, they can switch. They can use oxygen, and then sometimes they can switch and use something else like thiosulfate or nitrate as a part of their metabolic pathway. So that's definitely a big advantage if you can do that, because thiosulfate and nitrate are molecules that you tend to find in soil. So that's why sometimes you're driving by like Bolsa Chica wetlands and it smells like rotten eggs. Bacteria in the soil are metabolizing thiosulfate, making the gas, and it smells really bad, right? Okay, so we're going to focus in on the nitrate. What makes this a little bit tricky is we can't just throw in iron to just kind of label it so we can see, because with thiosulfate it's a lot easier. You can just throw in the iron and get that black color. For the nitrate lab we're going to do right now, you guys have to add the chemicals in order to get the color reaction to develop. So that's what you guys are going to do. We're going to add that. And what we're going to do is put this into a little bit of some context. The nitrogen cycle. So we didn't really get into this in lecture, but we kind of alluded that for a lot of the elements, carbon or sulfur or oxygen or nitrogen, they do get recycled, meaning one organism will use it one particular way and then release a product, and that will be used by a different organism. So a lot of our organisms are kind of connected, interconnected, in what we call a cycle. So we're going to draw out how our lab fits in to the nitrogen cycle, and I got kind of a hint from, oh boy, it's supposed to be a cow, but her cow was like way better than my cow. Oh, it still looks bad, and now it looks like an ant. No, it's like not good. She had like a way better. Yeah, it looks like a pig now. She did it out for me, and
it's an angry pig.
Yes.
It's a cow. It's like an
It's like, it shouldn't be like that. I'm trying to draw like horns.
Oh boy.
Huh? Yeah. That looks like an ant. That looks like an ant. And she even had like nice udders and everything. Me, not so much. All right. Oh, that's horrible. This is manure. And so what we're saying is down in the soil, soil is really rich in ammonium. Manure, dying plants, all that organic material usually releases ammonium into the soil. There are are some bacteria, and there was like a tiny, tiny bullet point about this on a PowerPoint, because we talked about nitrogen usage in the growth chapter, and we said peptone is really like the more popular choice, but ammonium can be used by some bacteria, because it does have nitrogen in it. So there are some bacteria out there where they'll actually use the ammonium, and strip the nitrogen from it, and they can actually convert it into nitrate. And then there are other bacteria, because nitrate also has nitrogen in it. There are some bacteria that actually can metabolize nitrate and convert it into nitrite. And it's usually bacteria that can do this. But there are kind of a more specialized, so the conversion of nitrate into nitrite, like metabolizing nitrate, there's a lot of bacteria that can do that. There's kind of a more specialized subgroup though that can metabolize nitrate even further. So I'm going to draw like 2 red arrows, where instead of just stopping at nitrate, nitrite, instead of just stopping there, they'll have some extra enzymes so they can keep metabolizing it further and further and further, and actually metabolize all the way into nitrogen gas. And not too many bacteria can do that though, so that's a little bit more specialized to be able to use nitrate and convert it all the way to nitrogen gas. In fact, there's kind of 2 main genuses that are notorious for doing that. Bacillus is one, and Pseudomonas is the other. And Bacillus and Pseudomonas are actually both soil dwelling. So it kind of makes sense because nitrate is something you find in the soil, and they can actually use it and metabolize it in order to make some energy, ATP. To complete our cycle though, what I'm going to do is loop it all the way around. So nitrogen gas is kind of hanging out in the air. And what there is there's kind of a subset of plants called legumes. Legumes would be like soybeans, usually like the bean-based plants. And legumes, they have roots with special nodules, like thick little pods on the roots. What makes them different from a lot of the other plants is in those pods, there's actually bacteria in there. And they share a really good relationship with those bacteria. It's a really good symbiotic relationship. And the reason why it's such a good relationship is because the bacteria that are hanging out in the little nodules of those roots, they can actually use the nitrogen gas in the air as their source of nitrogen. And again, it's kind of like a special group of bacteria that can do this. And these bacteria are called nitrogen fixing bacteria. And what they can do is they can just grab nitrogen from the air and actually reconvert it back into ammonium for the plants. So the plants love these bacteria because the bacteria are just pumping out ammonia. Ammonium is basically like plant food. So the plants, they like this relationship a lot. And so the two kind of hang out together, and then we kind of completed our cycle. So we have ammonium back in the soil, we have nitrogen gas in the air, and we have nitrate over here, kind of as like an intermediate. So that's how everything's kind of interconnected, but what we're going to focus on, we're not going to do the whole cycle. we're going to focus on this side. The nitrate usage. The nitrate usage. We want to see which organisms can metabolize nitrate, either just in the single step or metabolize it all the way into nitrogen gas. So that's the side that we're gonna kind of focus on. All right. So the way we're gonna do this is we're gonna break it up into two pieces. You guys are gonna do the first part of the test. We call it single step nitrate reduction. We call it single step nitrate reduction, which is kind of a wordy term. But what we're trying to say is, we do know that a lot of bacteria can metabolize nitrate. So we want to test for if they can metabolize nitrate, of nitrate, I want to say that. Test for metabolization of nitrate. And this is all happening during anaerobic respiration. So usually bacteria, a lot of times, if they can use nitrate, they'll use it towards the end instead of oxygen. And it says single step because what happens is they metabolize it in a single step. So it's metabolized in a single step. So in order to metabolize it, they have to have one enzyme, one enzyme. We mentioned the enzyme for thiosulfate. It was called thiosulfate reductase. Here it's the same idea. So they have to have a special enzyme called nitrate reductase. So in order to metabolize nitrate, you have to have that enzyme. You have to have it. If you don't have it, you won't be able to use it, which just kind of limits your options as far as what type of environment you can thrive in. So nitrate reductase will metabolize nitrate. And just as a reminder, so nitrate actually is in your broth that you guys inoculated. You guys inoculated, we actually called it a nitrate broth. So nitrate is our starting material. It's actually in the broth. In the broth, it's nitrate and basically just beef extract, and that's it. So the organisms don't really have a lot of options as far as if they want to grow. We want to see if they can specifically use the nitrate. You guys probably don't remember, there was a germ tube in there also when you guys inoculated. The germ tube, though, is not to trap gas from fermentation. The germ tube is in there to trap the nitrogen gas, right? So we're going to kind of use it to check for nitrogen gas. So, which is different from the gas made from fermentation, right? Because this is not a fermentation lab. All right. If your organisms can metabolize nitrate in just a single step, the end product that they'll make, it's not going to be a gas like hydrogen sulfide gas. It's just an inorganic molecule called nitrite. That's the end product that we end up with. So this is why you guys are going to add the chemicals yourselves in the back cabinet. You're going to add what's called, it says NO3A on it. We just call it A. Chemical A. And it's not important that you guys know exactly what's in chemical A, but it's basically an acid that's in the little vial. And what the acid is going to do is it's going to react with the end product. So it reacts with nitrate. So these two kind of react together. So the two kind of combine and react together, and then you're going to add a second chemical, and the second chemical is called just B, NO3B. And what NO3B is going to do is it's going to act like a catalyst, and it's going to catalyze a color change. So we're going to use it for color. All right, so the way this is going to work is we have A to kind of flag the end product. B is going to kind of give us the color that we need to see whether A and nitrite actually did come together and react. So with the two together, we're going to look for the color red. So if you guys add A and B, if you see red here, what's nice is you can just say, yes, I'm positive for single step nitrate reduction. I'm positive for single step nitrate reduction. So if you get red, after you add A and B, then you're done. There's nothing else you need to do. If though you add the chemicals and they stay clear, you can say, all right, I know I'm negative for single step nitrate reduction, but it requires a little bit extra testing, so I'm going to say you have to do further testing. So you're not exactly done. The reason why you're not exactly done is because there's still one other way they might use the nitrate, and that's with the red arrows, or I had like it continuing on a nitrogen gas. There's still a possibility of denitrification, so we don't know. I'm going to put it could be positive or negative for denitrification. So denitrification is different from single step, so we're going to draw it out on the other side of the board. So that's still in the running. Just because they're negative for single step doesn't mean that they can't use nitrate. There's still another possibility where they can use the nitrate. And the other possibility is what I have here on this side of the board. All right. So on the second part, you guys only have to do the second part of the test to your test tubes that are clear. So the second part will only apply to your test tubes that are clear. If you get red, you're done. You know, there's nothing else you have to do. So the second part is called the denitrification test. Denitrification is a little bit different because you don't just stop at that single step. This is a multi-step process. The organisms, they have to have extra enzymes to kind of be able to do the denitrification. And the nitrate is metabolized even further. And this is definitely kind of a lot more specialized. It'll help you out in your unknown project because not that many bacteria can undergo denitrification. That's a lot more specific. In denitrification, they still have to have the enzyme, nitrate reductase. But they don't just stop at the single step, nitrate reduction. They have to have extra enzymes in order to metabolize it even further. So, and I'm not going to write those enzymes in because it's not really important. But they would need extra enzymes. Nitrous oxide, and then finally, all the way into nitrogen gas. So we got some extra steps, And the bacteria would have to have an enzyme for each of these steps in order to complete the whole process. But the end product we would actually end up with would be nitrogen gas. Nitrogen gas. All right. So if you guys pull out your test tubes and you see a bubble in your Durham tube, that's kind of a big red flag that you're probably going to be positive over here for denitrification. In order to know for sure, though, you guys have to add the third chemical, which is zinc. So we're going to test it just to make sure, even if you see a bubble floating around in your Durham tube. What the zinc's going to do is the zinc is going to act as a catalyst. And what it's going to do is it's going to catalyze any unused nitrate into nitrite. All right, so to help clarify what that actually means, when you guys sprinkle in your little bit of zinc, we're going to have two possibilities. Well, the first one is you could be positive for denitrification. All right, let's say your organism is positive for denitrification. Let's say it can do all the steps we have written out on the board, and it can metabolize nitrate all the way into nitrogen gas. And it doesn't just stop at single step, it goes all the way towards the end over here, so it's positive. If you guys add your zinc, so if your organisms do this pathway that we have written on the board, you try to add your zinc. Is your zinc going to have any unused nitrate to interact with? No, right? So if your organism uses up all the nitrate and metabolizes it all the way to nitrogen gas, you try to add your zinc, the zinc's not going to be able to do anything because there's no unused nitrate. So there's essentially no unused nitrate for zinc. Zinc is not going to have anything to catalyze because your organisms used it all up. So it can't catalyze. And in your same test tube, we're adding the zinc on top of A and B. So keep in mind, A and B are still hanging out in your test tube. A and B, they're waiting to try to detect nitrite. That's their job. Remember on single step, we said we added it to detect nitrite. They're waiting around, but zinc isn't doing anything. It's not catalyzing. So A and B don't have anything to do either. So this is actually going to stay clear. So what's really confusing about this lab is if you get it clear after you guys add your zinc, that's actually a positive result. That's the most confusing part of the lab. If you are negative for denitrification, the opposite, let's do the opposite. If your organism can't do this pathway that we have written on the board, when you add your zinc, zinc will have plenty of nitrate to interact with because your organisms can touch it, so it's just sitting there in your test tube. So zinc will catalyze away, catalyzes NO3 into NO2. That's what it wants to do. And since your organisms never metabolize nitrate, it's just sitting in there, you add the catalyst zinc, it'll convert it into NO2. A and B are hanging out, waiting to flag the NO2 end product, and you will actually get red. So red is negative for denitrification. So the super confusing part is red is positive here, but red is negative on the denitrification part. The colors kind of flip-flop there. Yeah, so that is confusing. Does anybody have any questions about that part? I think it's confusing that it flips.
Yeah,
Flips
okay,
that
right.
way. So, and this is the key word here is unused. So Zinc is looking for unused nitrate to grab onto. If your organisms do use it though, zinc won't have anything to do, and so nothing will happen, and that's telling you you're actually positive for denitrification. We're going to kind of use the gas in the germ tube though, also as kind of like an indicator that we are probably going to be positive here. So we're kind of expecting nothing to happen. All right, so we're going to add A and B, look for single step, and then if it's clear, we'll test for denitrification, because denitrification is still a possibility. It's still in the running. Even if they're negative here, but let's say they're positive for denitrification. If they're positive for denitrification, then they are using nitrate, just not in a single step, but they are using it in this multi-step process. All right. Any questions about that? Jalen's like, I love this lab. I'm like, no. Especially about the color or... Oh, the other thing too is, so the zinc, remember we're adding that on top of A and B. So you guys in the clear test tubes is actually going to be A, B, and zinc. Everything together in the same test tube. So that's why I drew A and B are still kind of hanging out in there, waiting for something to react with. Nitrite. Okay, I'm going to give you guys your methods. It'll probably be good when you add the chemicals and you see what happens.
That wasn't it?
Yeah. I'm going to give you guys your methods, what exactly you're going to do. We switch to the document. Okay, go like this. Okay, we're zooming. We zoom back. All right. Okay, let's start at the beginning. So our methods, this is what we're gonna do for today. Everybody's gonna need 4 clean test tubes, which we have in the front. And we won't need any caps or anything, just clean test tubes. And then everybody's gonna need 4 glass pipettes. And I'm gonna have to show you guys how to use the pipettes because it's a little awkward. They're not the plastic ones that we've been using where we just peel away. Then what we're gonna do is we're gonna grab our organisms out of the incubator. The only thing that's a little bit inconvenient is we can't just add the chemicals directly to our test tubes that are sitting in the incubator. We actually have to siphon out a smaller volume. So with each of our pipettes, we're going to take out 2 mils of our culture and put them into the clean test tubes. So these are my 4 clean test tubes. And you're going to label, one's going to be the control, right? Remember, we have another one for E. coli, another one for P. aeruginosa, another one for M. luteus. And with each of our pipettes, we're going to put in 2 mils of organism into each clean test tube. So these are my 4 clean test tubes. We'll put the prospective organisms into each one. Then once we have that, we'll start adding chemical. So for the chemical, in the back cabinet I have A and B back there. We're going to put on some gloves. And what's nice is it's the same amount, so 5 drops of each. So we're going to do the single step test first. So we're going to add 5 drops of A to all 4 of your test tubes, even the control. So the control will also get 5 drops. And then we'll grab the B, and add 5 drops of that to all the test tubes, even the control. And then we're just going to gently shake it up and wait for the red to develop. So if you get the red, then you're done. You can say, yes, I'm positive for single step. My organism can use nitrate. It can metabolize it. I'm positive. If it's clear though, then I'm going to give you the zinc. And the zinc is like the last step. So for the zinc, we add, I call it a pinch. So you'll just take the cap off, you kind of then the dust kind of sprinkles out. We're going to add some zinc dust to our little test tubes, but only to the clear ones. And then we're going to just shake those up a little bit. So, and then we're going to look for red and clear again. The only difference is when we add the zinc, red is negative. Red is negative for denitrification. All right, so we're going to see if there's any unused nitrate in your test tube or not. So I need to show you guys how to use the pipettes, and then you can add your chemicals. Let me grab the pipette. Let's see. Okay.
Don't do that. Like no.
All right. The lab tech, he does try to set this up so every pipette in the conical tube is meant for 2 ml, a 2 ml volume, which is what we need for our test tube. These pipettes, you just kind of insert the pipette up into the little white plastic hole there. Then when you guys are pipetting up, so first of all, the other confusing thing about the pipettes is they do count in reverse. So at the bottom it actually says 2 mils, and then in the middle it says 1. There we go, middle says 1, and then it counts back to 0. So to get your 2 mils, you actually draw the fluid up to the 0 to get your full 2 mils, which seems a little counterintuitive that you're going to 0. But the other thing too, I wanted to show you our test tube racks. I didn't even realize this. So, because it gets a little clunky, because everybody needs 4 pipettes. So, your 4 pipettes. So if you want to, you can come up to the front. You can grab your 4 pipettes, and you can flip them. I notice your test tube racks have like tiny, tiny holes on the bottom. And you can put your 4 pipettes like that, because these are all autoclaves, so they're not like touching anything. And then you can just grab them one by one as you need them. And when you siphon up your little 2 mils, so you'll grab your cultures, you'll stick the pipette in here. You can actually use the wheel to draw up. There we go. Drop the fluid, and you'll draw all the way up to the 0. And then when you're ready to eject this into your clean test tube, you can just push down on the top and just kind of squeeze it all the way down. And then the fluid will eventually, this one's kind of slow, it'll eventually drain all the way out. So that's how you'll like eject it. You can use the wheel both ways, it's a little bit slower that way. And then when you're done with your 2 mils, there we go, it's taking forever. You'll take your pipette and you can just dump it off. You have a little tray. All right. And then you can go on and pipette the other test tubes. Okay, so these pipettes are in a drawer in the back for whatever reason. I don't know why, but the older ones that have the white caps like this actually work better than the other ones that are kind of naked, that are missing the white bottom piece. So you guys grab a pipette and you can grab a clean test tube. Yeah, you can share. Everybody doesn't have to have a pull out your
Oh, I don't know.
Oh.
Could you just put a bagel?
We need the test tube. Or just
Yeah, that's fine.
Technically.
Oh, no, that's the one? Yeah, the box.